Hello, good evening. My name is Richarda de Voer, and I'm an assistant professor at the Department of Human Genetics at Radboud UMC in Nijmegen, the Netherlands. And tonight I'm going to tell you about non-coding aberrations in mismatch repair genes that underlie a substantial part of the missing heritability in Lynch syndrome. By the end of this talk, you will have learned what the diagnostic route for the detection of Lynch syndrome in individuals with mismatch repair deficient colorectal and endometrial cancer is. You will know about the application of short and long read sequencing approaches to detect pathogenic variants in mismatch repair genes, and how you can use functional assays such as mini-gene assays to assess the pathogenicity of non-coding aberrations and their effect on splicing in the mismatch repair genes. In my talk, I will address Lynch syndrome, which is one of the most common genetic risk syndromes. And I will walk you through the quest for finding the missing heritability in Lynch syndrome. Because are all mismatch repair deficient tumors initiated by germline pathogenic variants? Or can we find non-coding aberrations in mismatch repair genes? I'll end my talk with a summary and a short future perspectives on what I think the next steps should be. But let me start by explaining you the genes that are involved in hereditary colorectal cancer and polyposis syndromes. I've been showing this graph already for about a decade, and we know in general that about 25 to 30% of colorectal cancer cases are probably due to a genetic predisposition. There are about 16 genes now known, and all of them cause high penetrant forms of hereditary colorectal cancer. However, we also think that there are still undiscovered hereditary causes, and you may find them in familials in which there's a strong aggregation of colorectal cancer and polyposis, but also sometimes in very isolated cases, for instance, colorectal cancer at a very young age without a clear family history. In my talk, I will focus on Lynch syndrome, which is caused by germline pathogenic variants in the genes MLH1, MSH2, MSH6, PMS2, and EPCAM. These genes have already been discovered about 20-25 years ago by basically a very targeted sequencing in the pre-NGS era, so in the pre-next generation sequencing era. More recently, in 2009, EPCAM was added to the genes that cause Lynch syndrome, and all of them cause a non-polyposis form of colorectal cancer. So in particular, what is Lynch syndrome? So it's one of the most common forms of genetic tumor risk syndromes, and it's caused by these germline pathogenic variants in the mismatch repair genes. When an individual has such a germline pathogenic variant, they have a high risk to develop colorectal cancer. About 50 to 70% of individuals will develop a colorectal cancer or multiple during life. Women have a higher risk to develop endometrial cancer. Approximately 40 to 60% of the women will develop an endometrial cancer. But we also know that there's a higher risk to develop extracolonic cancers, for instance, in the bladder and in the stomach. Usually these cancers also develop at a younger age of onset, especially before the age of 70 years. So basically before the cancers that develop that we call sporadic. So the mismatch repair genes are involved in a mismatch repair machinery, and they repair basically mismatches in our DNA. So DNA polymerase synthesizes the DNA, and this is not always perfect, and then the mismatch repair complex comes into action. So they repair DNA mismatches, either single nucleotide variants, or they repair nucleotides that have been wrongfully inserted in mononucleotide repeats, or if a base pair has not been inserted in a mononucleotide repeat. So when the mismatch repair complex is active, these are repaired, and you will have stable microsatellites. However, if the mismatch repair machinery is not active, these mismatches in the DNA are not repaired. And also in microsatellites, these will be either shortened or longer. and this will result in microsatellite instability. The accumulation of both single nucleotide variants and this instability at microsatellites is usually the first step in the tumorigenesis. It inactivates tumor suppressor genes and it may activate oncogenes. And this is also what is used in the diagnostics of Lynch syndrome, in particular this microsatellite instability and the absence of proteins in the mismatch repair machinery. I'm going to walk you through the current diagnostic route for the detection of Lynch syndrome as we nowadays use it in the Netherlands. In principle, if a colorectal cancer or endometrial cancer developed before the age of 70 years in an individual, the pathologist requests an immunohistochemistry of the mismatch repair proteins. If the immunohistochemistry is normal, meaning there is expression of all 4 mismatch repair proteins, it is very likely that the colorectal or endometrial cancer developed sporadic, or there's an involvement of another genetic tumor risk syndrome. However, in the absence of one or more of these mismatch repair proteins, Lynch syndrome is one of the syndromes that is considered. Then there's the next step. If the tumor is MLH1 deficient, we will have a look at promoter hypermethylation, because if the promoter is hypermethylated in the tumor, we usually also think it's a sporadic tumor that has developed, or there's involvement of another genetic tumor risk syndrome. In case the MLH1 promoter is not hypermethylated, these tumors are also eligible for germline testing of pathogenic variants in the mismatch repair genes, like is the case if MSH2, MSH6, or PMS2 are absent. At that time, after genetic counseling, a germline test is offered to the patient, and we will test for germline pathogenic variants in this mismatch repair genes. Usually, we only look at the coding regions of these genes, and if a germline pathogenic variant is identified, the patient is diagnosed with Lynch syndrome. This is approximately the case in 55% of the individuals that are referred for genetic testing, meaning that in over 45% of these cases, we do not find a germline pathogenic variant in one of the mismatch repair genes. These are called the unexplained mismatch repair deficient cases, or also known as Lynch-like in literature. These Lynch-like cases, or individuals that have an unexplained mismatch repair deficient tumor, are a puzzle for clinicians, because we do not know what their cancer risk is, what should be the surveillance to offer, how often should individuals undergo a colonoscopy, what is the risk for their family members, can we offer them any surveillance for the future. This is a big puzzle for clinicians. We do know that 2 somatic inactivating events can also cause mismatch repair deficiency, meaning that in somatic tissues, the mismatch repair genes are inactivated and therefore causing microsatellite instability and thus also a sporadic cancer. While we already know this for about 8 years, it was until recently not known how often this occurs. So a very talented PhD student in the group, Lisa Else, she hypothesized that somatic non-epigenetic mismatch repair gene aberrations underlie most mismatch repair deficient, or as we call them, Lynch-like tumors. She analyzed both germline and the somatic coding aberrations in the mismatch repair genes in 2 different cohorts. The first cohort was composed of 306 individuals, in which the MLH1 promoter hypermethylation was excluded. After analysis of the germline DNA for mismatch repair gene aberrations, she identified that in approximately half of them, a germline pathogenic variant was found. For the remaining part of this cohort, 125 tumors were available to perform somatic coding analysis of the mismatch repair genes. In a second cohort, which was already pre-screened for MLH1 promoter hypermethylation, and for germline pathogenic variant, and in both cases this was excluded. 123 tumors were available, also to perform somatic analysis of the mismatch repair genes. So what we normally do is we capture the mismatch repair genes using single molecule inversion probes, and we perform next generation short read sequencing of the genomic DNA extracted from both normal and from tumor tissues. And we look at pathogenic variants that occur in the tumor that are absent, for instance, from the normal tissue. What she found was that basically the majority of unexplained mismatch repair deficient tumors have two somatic mismatch repair mutations. As you can see in the figure, in both cohorts, we find over 80% of the tumors that have two somatic events in a mismatch repair gene. This seems to be a bit higher for MLH1 than for MSH6 and PMS2, but on average, 87.3% of unexplained mismatch repair deficient tumors presented with this somatic mismatch repair deficiency. In addition, in this cohort, we looked at the age at diagnosis of these patients, and it seems that individuals that have a somatic mismatch repair deficiency have a higher age at diagnosis compared to those that have Lynch syndrome. As you can appreciate from this picture, individuals with Lynch syndrome are usually below 50 years of age, whereas individuals that have a somatic mismatch repair deficiency, this increases with the age. So once the germline pathogenic testing of the mismatch repair genes was negative, if after that you perform somatic testing of these mismatch repair genes, we can explain in over 85% of them the cause of their mismatch repair deficiency, basically by two somatic hits in one of these genes. This still leaves a group of unexplained mismatch repair deficient cases. This is a bit less than 15%. They either have one or no somatic hits in the mismatch repair gene, and there's also an absence of pathogenic variants in the germline. Still, again, the same questions are valid. So what is their risk for cancer? What surveillance should we offer them? And what about their family members? And although less than 15% seems like a very small percentage, it does have a big impact. Several of these families basically look like this. So they have an unexplained mismatch repair deficiency, but they developed cancer at a very young age. For instance, the index case in this pedigree was 33 years old when he developed a colorectal cancer. His mother developed colorectal cancer at the age of 47 and an endometrial cancer at the age of 50. And also multiple family members were diagnosed with the same tumor types at a very young age. All of them showed microsatellite instability and loss of mismatch repair proteins. However, thus far, we could not pinpoint what the reason for this mismatch repair deficiency was. So therefore, another very talented PhD student in the group, Iris de Paske, hypothesized that non-coding aberrations in mismatch repair genes underlie this form of Lynch-like syndrome. And she started with the design of targeted long-read sequencing of the MMR genes. So again, I depict here a gene and the regions that we normally sequence when we perform short-read sequencing, so only the coding parts of each gene. And she designed long-range PCR amplicons that cover basically the entire coding and non-coding parts of the mismatch repair genes, including EPCAM. So what does this look like? When you perform these long-range PCR products, we also take into account the introns, in which we think that we can identify structural variation or single nucleotide variants that lay deep intronic, but we can also discriminate between variants that are present in the pseudogene instead of in the targeted gene in which we want to identify the pathogenic variants. To give you the first example, she designed multiple amplicons to cover both EPCAM and MSH2 with overlapping segments of about 100 kb. The smallest fragment for these two genes was approximately 10 kb in size, whereas the biggest fragment was close to 18 kb in size. We also designed these amplicons for MSH6, MLH1, and PMS2. And for PMS2, we particularly took into account all the different pseudogenes and that we would reduce the chance of also amplifying in these long-range PCRs any pseudogenes. So we designed long-range amplicons with a 1 kb overlap, followed by long-range PCR with optimized protocols to amplify each of these amplicons. Subsequently, we performed equimolar pooling of these amplicons, and we performed circular consensus sequencing on a PacBio sequel2. After sequencing and consensus calling, we performed mapping of these high-fidelity reads and variant calling. We also performed this on some positive control samples, so meaning previously identified pathogenic variants. And in this example, I show you some of the positive controls for PMS2. In the upper graph, you can see PMS2 and the entire locus that is covered by our long read sequencing. In the red box, you can identify reads that span exon 14. They are approximately over 1,500 bases in size. In the bottom left, you can see a single nucleotide variant that was identified. And in the middle, you can see a more complex variant, a del-in of multiple nucleotides. Then on the right, again, 2 deletions spanning exon 14, whereas we always thought that this would be the same deletion. We observed in 2 different samples, 2 different deletions, and we now also can perform accurate deletion mapping of larger deletions. So back to our study, we performed this targeted long-read sequencing on multiple individuals with Lynch-like syndrome. We were able to collect 32 individuals that developed a mismatch repair deficient colorectal or endometrial cancer. No MLH1 hypermethylation was identified and no coding germline pathogenic variants were observed. And somatic testing revealed only 1 or no somatic event. We performed our long-range PCRs and performed long-read sequencing. And after mapping and variant calling, we performed subsequent filtering to identify any potential pathogenic variants. Because we are looking for germline variants, we only select those variants with a variant allele frequency of over 20%. And we remove any variants that are more than 0.1%, that have a frequency of more than 0.1% in public databases such as EXAC and GNOMAD. Furthermore, we make the discrimination between variants that lay in the exons and non-coding variants. And specifically for non-coding variants, we set the threshold a bit higher for the variant allele frequency to circumvent any slippage in homopolymer sites. Every variant was assessed for potential effect on splicing using Splice AI, and all variants that remained were visually inspected using the integrative genome viewer. This resulted in a potential pathogenic variant in 9 out of these 32 cases. Deep intronic invariants were identified in 5 cases, and in 4 cases we identified structural variants of multiple bases. And subsequently, we performed co-segregation analysis in families where this was possible, and we performed functional testing. This is the overview of the various variants that we identified in these individuals. On the left, you see that we identified variants in MSH2, MLH1, and PMS2. In some individuals, we identified 2 deep intronic variants. Either these were single nucleotide variants, or in individuals, we identified the insertion of an allo repeat of over 350 base pairs and single nucleotide variants. Then there was a case in which there was only an allo insertion. In MLH1, we identified a case in which there was a large intronic deletion, also single nucleotide variants. And in PMS2, we identified the same single nucleotide variant in 2 individual cases. So what can be the consequence of a deep intronic donor site introduction? Here I show the example of one of the deep intronic variants that we identified in MLH2. And using in silico prediction tools, we noticed that this A to G change activates a cryptic acceptor site approximately 190 bases upstream. By activation of this cryptic acceptor site, we predicted that there would be a pseudoexon included in the transcript of MSH2. Interestingly for us, from both the index case in which we identified these variants and 2 family members, we were able to gather RNA derived from whole blood, and we tested for the insertion of this pseudoexon. And as you can see in the graph below, in the control, we were only able to pick up a fragment of 485 base pairs in size, but in the 2 individuals on the right, who are all indicated as Lynch-like, we could identify a fragment of 673 base pairs, indicative of a pseudoexon inclusion. We also performed expression analysis of MSH2 in one of these individuals. And compared to controls, we saw that there was indeed a reduced expression of MSH2, which we could rescue by adding cyclohexamide, which is an indication that at least one of the 2 alleles undergoes nonsense-mediated decay. We performed segregation analysis in this family, and all family members that were available for genetic testing presented with this germline pathogenic variant in MSH2. And this solves basically this family, and now from being a Lynch-like family with unexplained mismatch repair deficiency, they fall into the category of Lynch syndrome, which means that we can provide them optimized surveillance and also targeted mutation analysis in any family members that may want to get tested. However, not for all the variants that we identified, we had access to either material from the individuals to perform mRNA analysis or segregation analysis. And for those, we performed functional analysis by the use of mini-gene splice assays. And in principle, we mimic both the wild type and the mutant variants in plasmids that contain the row exons. So basically, our gene region of interest from wild type is cloned in one vector, and subsequently in another vector, our mutant region of interest is cloned. We then transfect cells, and we perform cDNA analysis. And if the mutation has an effect on splicing, for instance, introducing a pseudoexon or exon skipping, we would see a different, we would see this on an electrophoresis gel as a difference in size. And after sequencing, we can pinpoint exactly if there is a splice site defect. So for each of our variants, we performed this analysis. And I give you the example here of one of the variants in intron 3 of MLH1. And as you can appreciate in this picture, we observed a larger band in the mutant construct versus the wild type construct. And after sequencing of this band, we observed a inclusion of a pseudo exon in this case. Together, we observed that in 5 of our variants, a pseudo exon was included, or an exon 2 was skipped. We could observe altered splicing, meaning that in 6 of our cases, we identified that these non-coding aberrations functionally showed an effect in our mini-gene and promoter assay. In 4 of the cases with an MSH2 variant, we saw that this led to pseudo-exon inclusion or premature stop, or exon skipping. And in 2 out of the 3 individuals with an MLH1 variant, we observed altered splicing and a premature stop codon, or pseudo-exon inclusion and a premature stop. 2 variants remain variants of unknown significance, as we were unable to determine whether these are really pathogenic. And for the other ones, our results pinpoint that they're likely not pathogenic. So in summary, the majority of individuals with unexplained mismatch repair deficiency, or so-called Lynch-like, have somatic inactivation of one of the mismatch repair genes. A substantial proportion of individuals with Lynch-like syndrome, approximately 90%, can be diagnosed with Lynch syndrome because of a germline non-coding pathogenic aberration in a mismatch repair gene. We think that diagnostics mismatch repair gene panels should be expanded for known intronic pathogenic variants to increase the diagnostic yield for Lynch syndrome. However, for those cases that remain without a genetic diagnosis, and in our case, we still have 80% of them to solve, we think that cases that after both germline and somatic routine diagnostic analysis, it remains of interest to perform germline sequencing of the complete mismatch repair gene loci, for instance, using long-read sequencing approaches, also in a genome-wide context. Also, with our targeted approach, we have probably not been able to identify every variant in these mismatch repair genes, and hopefully in the near future, we can identify new variants. I would like to acknowledge specifically Lisa Else and Iris de Pasque, who have done most of the work that I've presented here, but also my collaboration with Nicolien, Marjolein, Shella, and Arjen has been of high importance for these studies, and all other people listed here to provide samples and useful discussions, and our funders. Thank you very much. I would be very happy to answer any questions that you may have, and you may put them in the Q&A box, and I'll be happy to provide an answer. Thank you very much.